Dr. Walsh here, John Walsh, call me John. All right. So this will be the second lecture for the week of June 17th. Okay. So again, the concept is you're going to have two topics that you gain intellectual control over. Okay, that you learn about every week. All right. So I sent you the video that described what you needed to do for the first content. And again, when we do content, it's going to be here. And we're going to go back. Okay, and it's going to be so that I clicked on the readings and here, and then it's a discussion. Okay, readings, discussion. You go back and forth with this. Okay, each week you're going to do one reading and quiz, and then an associated discussion, and then the second part of the week you do the reading quiz and associated discussion. Okay, now the quizzes, by the way, they are open, so you can work ahead of those. Okay, the discussions, however, are not. They get released every week, so you'll see the discussion board open up for you Tuesday morning, first thing, right after midnight, and then they'll remain open for an entire week. Okay, so then you have all the way to the following Monday, and then that Monday at midnight, then it closes down. Okay, remember with the discussion, one substantive post, 3 posts that you comment on what your friends have said. Okay, all right. So back and forth with that. Okay, awesome. All right, and we'll keep you updated on these other assignments. Okay, as they come up. All right. So let's just focus on the content. So we're going to go back over here. Okay. And I'm not going to download the syllabus again, but you can go ahead and do that. So this right here, again, this is the week of June 17th. So I went through this lecture last time, and now we're going to go into this lecture right here. And this is the study of epigenetics. Again, I understand you're not a biologist. Do not get caught up in vocabulary. So, epi means outside, and genetics. Okay, so what does that mean? Okay, so all of you know about your chromosomes and your DNA, okay? And you inherit copies from your mom and your dad, and that makes you who you are, okay? It starts out with a sperm and an egg, and lo and behold, you have all these different cells that we have here, okay? However, every single cell in your body has the exact same set of chromosomes, okay? And those are the ones you got from when you became a fertilized egg, alrighty? And so it's a cookbook, okay? It's many chapters in that cookbook, okay? And but the way cells work is you just go to each chapter, and there could be thousands of recipes in that chapter, and you just pick and choose which recipe you're going to read out, okay? Which recipe you are going to use to as the design for manufacturing a protein, and that's what it's all about, okay? And then those proteins interact to do things in the cell, make cells divide, make nerve cells send dendrites out. Any process in terms of how cells work, okay, all starts with the assembly and then the kind of timing of interaction of all these different proteins. And this is not a static thing where it just happens once and you're done. This is a dynamic thing happening just at light speed in all your cells all the time. Okay, all right. So now we have to choose, which recipes that we need to read out, okay, to make the new proteins. All right, and so those recipes are called genes, and so developmentally, okay, one gene being read out to make a protein causes kind of like a domino effect that will cause other genes to be read out based on the environment that cell's growing in and things like that. And next you know, you have a kidney cell, you have a vascular cell, you have a brain cell, and it's all again coming from the same exact cookbook, the same set of chromosomes. Okay, and it's just a matter of these what are called epigenetic tags. And we're going to be discussing just one, which is called a methyl group, a methylation. But there are different types of tags. There's a thing called an acetyl group or acetylation. There's another type of tag. And again, the way I look at it, it's like having just a bunch of Post-its. Okay, so here's my Post-it right here. Okay, and let's see here. I'm kind of looking around for it. Okay, so this is not a book, but we can pretend this is a book. It's a binder, and so this is my DNA. Okay, and so how do I know which proteins I want to read out? Okay, so here is a methylation tag, and I'm going to stick it, okay, right here on my book, and I'm going to stick it right on the recipe. Okay, and so there is I'm coming backwards here. There it is. See my Post-it note? That is an epigenetic tag. Same book, but now I'm only going to go here and study that recipe to make that protein. Okay, so that's pretty much how it works. Okay, alrighty. So you can watch this short video, well done, about this concept of epigenetic tags and how it works. Okay, and then we're going to do a really fun interactive work through of this amazing website. Okay. So in this particular case, they talk about diet. Okay, that's an environmental influence, and they talk about psychological stress, another environmental influence that will determine which recipes are going to be read out to make which proteins. Sometimes in a good way, adaptation, sometimes in a bad way that causes a cascading series of events that can lead to vulnerability and disease. All right, so that's that concept. All right, so then you come down here, okay? Again, this is student view. I'll be switching back and forth to what my view looks like, okay? And so you see how the little hand lights up. So we've made it so that this will take you to the website. Also, down here, this is the link to the same website, okay? So we're just gonna click on this, okay? And it takes me to this really cool interactive website, all right? So you've already watched that video, okay? And now you click on this right here, okay? And you watch this second video, all right? Short video, it was 1:47 seconds long.
Contains the instructions for building all the parts of the body. But DNA is only half the story. The DNA in our bodies is wrapped around proteins called histones. Both the DNA and histones are covered with chemical tags.
There's
This
the tags.
second layer of structure is called the epigenome.
All right, so when the DNA is all wrapped up around that histone, it's like a binder clip on your cookbook. You can't get in there to read the recipe. All right, it's only once you remove the binder clip. Okay, all right. So and removing the binder clip would be like either adding or removing one of these epigenetic tags. Then the DNA unwinds and you can get in there and find the recipe to make the protein that you want, okay? All right, so I'll show you right here.
The epigenome shapes the physical structure of the genome. It tightly wraps inactive genes, making them unreadable.
So you see it's all tightly bound. So we have a special set of proteins that will go in there and say, hey, I want to make a copy of that recipe, okay? Because I want to bake a cake, right? But you can't get in there. You can't find the recipe because it's all bound together, right? But it's only when you change the tag that you get access to the...
It relaxes active genes.
There we go.
Making them easily accessible.
All right.
Different sets of...
So you get the concept. I'm not going to watch the entire video with you. Pretty amazing stuff, okay? All right. So that's the first part, okay? Then we go back over here. We're going to go back, and we're going to look at controlling the genes. Now, some of you may or may not have Flash, and that's what this interactive exercise is about. You can download Flash, okay? And you just, I have allowed it right here, all right? All right, so now I have the interactive exercise going on here. Okay, so you kind of read through this, okay? And it's talking about a way to monitor expression of gene activity. And scientifically, one of the real kind of revolutionary changes in science at the cell level to monitor which recipes are being read is scientists developed a method. In fact, the scientists that developed this method down at UC San Diego identified a protein called a green fluorescent protein. Okay, it's actually the protein that causes luminescence in biology. Okay, bioluminescence. Okay, so it was identified in squid populations. Okay, and so what he was able to do is you're able to put this green fluorescent protein and put it right next to a recipe or a gene of interest, something that I'm really, really excited about looking at. Maybe it's a gene that causes cancer. Okay, so then every time that gene that causes cancer, okay, gets expressed, so we can make the message, the recipe, or we can make the protein. Okay, and that's the key concept. So when I read the gene, then I make the messenger RNA, which is the recipe that then makes the protein. Okay, so every time that protein is made, it has a little fluorescent tag. So this is how I find out if things are increasing or decreasing. Okay, so we're going to show you what happens here. So I'm going to take this little dial. Okay, we see right here. All right, so I just closed the book. All right, put a big binder clip on it. You got no recipes being made, and as a result, no proteins being made. Okay, so it's my cookbook. I can't find the recipe. I'm not making a cake. Okay, so now, all right, this is the same DNA in all of your cells, same cookbook, but now because of changes in my environment, okay, any change in my environment, okay, whatever I'm having for breakfast that morning, okay, how stressed out I am. Those are the examples we're talking about. It's hot outside, it's cold outside, any kind of change psychologically, things that happen to you, okay? All of a sudden, you see right here, is because of the change in these epigenetic tags, I'm able to open up my cookbook, unwind it, okay? And as I unwind it, what are we seeing here? This is like the little Xerox machine. I'm making the recipes, okay? So the DNA is in here. We make the RNA, okay? All right, and this is called messenger RNA. And you'll see I'm starting to make more and more and more and more copies. And from that, then all my little cooks that are out there, okay, can read these recipes and make the proteins. And now I'm able to find out what proteins are being made because they're tagged with this green fluorescent protein as well. All right, so it's a really neat scientific method. All right, so you get a chance to kind of look through this a little bit right here and have more detail. That's part 2. Have fun with that. Okay. All right, so this is about changes in regulation of your epigenetic tags. Okay, so this goes over things like I was talked about. So we start here, okay, from stem cells, and then we grow to all kinds of different types of cells in our body. Okay, and it talks about the changes that happen developmentally. All right. All right, so you have all these developmental changes, a sequence of events, and you see it's like, as you see here, this domino effect where cells are interacting. That's what that's all about, and that's how we become who we are. Okay, this tells you a little bit about the signaling that happens. Okay, so an outside signal, so one cell or some weird crazy thing happens to us environmentally. Okay, something gets into our blood, right, and now the cells need to sound an alarm. They maybe send out hormones or whatever. The hormone binds to a surface receptor, and then that goes down there and affects this epigenome, affects which parts of the cookbook are going to be read, okay, which recipes are going to be produced, okay, and that's the way that works, okay, and this kind of goes through this whole thing. All right, so that's the second part. Straightforward stuff. You're not biologists, but just have fun with this, okay? This talks about heritability, okay? Alrighty, and kind of amazing studies that are in terms of the genes that are passed on and the concept, okay, that things you do in your life, all your environmental exposures, okay, whatever is at the height of environmental exposure, when you reproduce, okay, those switches, those post-its on your DNA are passed on, okay, to your offspring, which is scary if you think about it. It's amazing, okay? So that's what this kind of goes through right here, okay? And again, I want you to think about this a little bit. So it shows you a woman right here, okay, who's smoking a cigarette during different sequence of epigenetic tagging in the developing fetus, and how that can then impact the child for the rest of its life potentially. Okay, that's what that's all about. Okay, so that's inheritance. Okay, this right here talks about, again, the genes that you do inherit from your mom and dad. So here's dad, here's mom. You see, again, the DNA that is donated to the fertilized egg, right? And they have all their tags. So here's all of dad's tags from everything that's gone on in his life and everything he inherited from his parents and grandparents and all that. It shows you how it's all passed along, okay? And then when we look at moms, okay, there is a dominance effect. So it just works out that one set of genes from mom and one set of genes from dad will dominate, okay? And it's random. You think about the thousands and thousands of genes, okay? One dominates from dad, one dominates from mom, and that's why some traits of your kids are a lot like you, and some traits of your kids are a lot like your spouse, okay? Alrighty. And again, it goes through all the different aspects of this imprinting from the source of your genome, the source of your DNA. All right, that's what that's all about. Okay, this talks a little bit about the insights that you get when you look at identical twins. Now, with an identical twin, remember, it's one sperm, one egg. Okay, so the identical DNA, the identical epigenetic tags. Okay, the post-it notes all throughout that were inherited from you and your spouse. Okay. And then what happens is the cells divide and boom. And now they're continuing to divide, boom, boom, 2, 4, 6, 8, like that. Sorry, my math was wrong. 2, 4, 8, okay, 16, 32, all the cells are dividing. And now unique environments that they are exposed to, okay? For the most part, in the womb, it's going to be identical. But the second you give birth, boom, now different environmental experiences, okay? So we can watch and see what happens by studying identical twins and looking to see the epigenetics of development, the epigenetics of disease, all these kind of things. So it kind of goes through this and it looks at different things, okay? So we look at things like height, and we look at identical twins and fraternal twins, and we see the pattern of inheritance and what's most important. So just kind of rifle through that. Really cool stuff. Okay. This one right here, okay, now you're going to manipulate the environment yourself, okay? So are you nurturing as a mom, okay? And that's what this is all about right here, okay? And by looking at changes in nurturing, and this describes this right here, you can look at changes in terms of the epigenetics, okay? All right, so we go right here.
Deep in the brain of a newborn rat pup, methyl molecules, green, silence the GR gene. When it's active, the GR gene produces a protein that helps the body relax after a stressful event.
All right, so GR stands for glucocorticoid receptor gene, okay? So I'm going to lick once. I'm going to lick twice. So I'm clicking. I brought the rat over.
It looks like you had better things to do, since you didn't lick and groom your pup very much. Its GR gene is still inactive. Your pup will have a hard time relaxing after stress.
Yikes.
Your pup's GR gene will most likely look like this for the rest of its life. In fact, the amount of nurturing you gave to your pup will have a major impact on its adult personality.
Wow. Sorry about that, first child. Okay, let's try again. Okay, so I'm gonna have a second child, and I'm gonna do much better. So I'm gonna hit go. I'm gonna grab my rat here, and I'm gonna lick, 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 lick my pup, and we'll see what happens.
It looks like you had better things to do,
Oh!
since you didn't lick and groom your pup very much. Its GR gene is still inactive. Your pup will have a hard time relaxing after stress.
Well, that really
Your pup's GR gene will
messed up.
most likely...
Here, try again. Oh, you know why? I'm licking the DNA. That doesn't work. Watch this. You can say the same thing.
It looks like you
Okay,
had
wait a
better
second.
things to do.
I can lick the pup. Oh, look, see how his ears move? Look at my DNA opening up. Boom, 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 boom. So that was me being lame. But now I got it figured out.
Wow, you're an attentive rat mother. It's surprising your pup has any fur left on its
It's like my
body.
third child to figure
Your
it
licking
out.
and grooming has
Instead of
seriously
nurturing my
activated your
child, I
pup's
was
GR
nurturing a
gene.
book.
Your pup will have an easy
Okay.
time relaxing after stress.
Let's do it one more
Your
time.
pup's GR gene...
I'm an amazing dad now. And look at how the DNA opened up. All right, so that's a good thing in this particular case.
Your licking and grooming has made your pup's...
All right, so you get the concept. We can look at things now like nutrition. All right, like I said, what you are what you eat. Okay, you've heard about that. Okay, so it actually goes in, will affect your DNA. Okay, you've heard if you've anybody ever had a child, you know the doctors were a little bit freaked out on making sure that you had enough folic acid because that affects methionine that puts the methyl groups on your DNA. Okay. And when you don't do it right, you have trouble. Okay, folic acid has a whole set of metabolic effects, so that's not the only reason. All right, so so this goes through all these different types of things. Okay, alrighty, and how diet can change things. All right, this is an interesting historical story right here. Okay, all right, so check that out too. All right, so and then it ends with brain development. Okay, and we can listen to this video. It's 7 minutes long. Somebody who studied brain development and things, a link between child abuse and suicide risk based on licking the rat pup or based on whether or not you had a nurturing childhood. All right, that's what that's all about. All right, so please interact with that. And then we come over here to the actual article. Okay, and once you've gone through the interactive exercises, so I'm downloading the article, there I am right there, then this will become much easier. So you just read through, so we're looking at what? The epigenome, okay? Again, these are the post-it notes to my DNA, okay? And we're gonna look how that affects aging and age-related disease, okay? All right, and what is the environmental impact? The environmental impact is your life's stress, okay? And the release of this hormone during stress, which is called a glucocorticoid. It is the cortisol released from your adrenal gland. All right. All right. So we come down here and we're going to look at this concept. Okay. So we last time learned about our brains interacting with our body. All right. So our brains, the hypothalamus is told to bring on board the body. Okay. Our higher cognitive centers interact with the hypothalamus. Remember, the amygdala drives the hypothalamus, okay? We have our frontal cortex and hippocampus that modulate the amygdala, all right? If the hypothalamus is in overdrive, it then interacts with the pituitary gland stuck at the base of the brain. It releases a releasing factor that goes to the adrenal gland that causes the release of glucocorticoids. Cortisol, okay, is a glucocorticoid, all right? And then that alters the post-it notes in your cookbook. That alters epigenetic tagging of all of your DNA and all your cells, okay, that are exposed to it. And then there's interactive effects again as well. Alrighty, this based on stress can accelerate the aging, the pattern of genes that are expressed, the patterns of proteins that are produced, okay? And you can measure this, and that's what this is all about. So I want you to just not worry about things, okay? And as non-biologists, kind of read through this and kind of enjoy the ride, okay? But we're going to talk about things like, God, how I live my life and how that impacts me later on in life in terms of cardiovascular disease risk, cancer risk, dementia risk, okay? All this, okay? And is it the only thing? No, okay? We saw from the video with this one concept of telomere shortening, and that exposes all of our chromosomes to a greater likelihood of mutation, and then the epigenetics do it as well. Okay, all right, so this goes, again, through a series of studies that you can maybe use someday, these papers, all right? And it talks about different types of stress. Okay, so there's perceived stress, there's early life intrauterine stress during childhood. Okay, there's work stress. Okay, there's chronic and excessive stress, some childhood trauma. Okay, these are all different things that happen that put you at risk for cardiovascular disease, brain vascular disease, development of cancers. Okay, on and on and on and on. Okay. As you're going along, they're going to be, you might even write down these acronyms. So the hypothalamic to pituitary, so that's the brain, to the pituitary, to the adrenal axis that releases cortisol, okay? This right here is the hormone, okay? Adrenal corticotropin hormone that is released by the pituitary that goes down to the adrenal, telling it to release the glucocorticoid or cortisol, okay? So just kind of read through this, get familiar with it, okay? This right here, the glucocorticoid, cortisol, the most famous, interacts with cells through this glucocorticoid receptor. That's what that is right there, okay? And this glucocorticoid receptor then will go into the cell, and it will modulate whether or not the DNA gets methylated, okay? And so that's what they're talking about here. And this is a particular site in the DNA, and they talk about this throughout this article, that is either methylated or not methylated, okay? So they're focusing in on methylation in this article, okay? So then they talk about what happens here with the methylation of this site in aging and during different aspects of a person's age. Is it a stressful aging? Is it a malnourished aging phenotype? Okay. So I'm not going to read through each of these articles, the entirety of this article. I'm sorry, but you will find, okay, that this is very, very, very intuitive after you hold back and just read it for fun and don't get caught up in the details. Okay. All right. So this is a figure that kind of goes through some of what's going on. Okay, aging by itself affects the expression of all these proteins that are all involved in the response to stress. This is your glucocorticoid receptor, but also right here in your methylation processing, and that's all this is right here. And I'm sorry it has all these acronyms, but this is the methylation processing that is impacted by aging. And then there is this interactive effect with stress, an environmental factor right here. So it's, again, it's sometimes these figures can be more complicated than they should be, okay? But in the end, this is your DNA. So here's your nucleus. This is the DNA. Remember, the cortisol comes down. It interacts with this receptor, goes into your DNA, all right? And it's going to then affect which recipes need to be read. Messenger RNA is produced, and what proteins are going to be made. Right? So these are the methylation changes, the epigenetic aging changes. Okay? And then these are all different parts of the chromosomes that get modified that, if are maladaptive, can cause all kinds of aging-related diseases. And a phenotype means as what it looks like. So the phenotype of cerebrovascular or cardiovascular disease is all the gunk that gets built up inside of your vasculature, okay, the atherosclerosis. All right, so what's I think very interesting about this table, okay, is looking at the effects of life stress on these phenotypes, okay, these different readouts, okay, of aging, okay. So choose your stress, okay? So we have combat, childhood trauma, PTSD, okay? It talks about the finding right here, all right? It talks about what was assayed, okay? So, and you can go through all this, okay? We look at right here, depression, PTSD, okay? We look at the groups. Right, we'll scroll down a little bit more, okay? How about this? This is perceived racial discrimination, okay? Parent-child conflict. All these were looked at, okay? And changes in the epigenetics. So these are all life stressors. So it's really, really kind of interesting about the environment. All right, and then you're all done with this. This is the wrap-up. It talks about harsh parenting. It talks about lots and lots of different things, okay? All right, cool. So I'm going to get out of this, all right? What I'm going to do now is I'm going to exit the preview because I want to be able to show you the 2 assignments then that you have to do for this. So we're going to exit the preview. All right, continue. I had laughed that I was linking my DNA instead of my rat pup, okay? I didn't read the directions, okay? All right, so now this is my view, so it's a little bit more expanded, okay? So back to the readings, okay? And back to this part right here, that's the second lecture. And now you'll see is what you have is a quiz. So I clicked on the quiz, okay? I'm gonna hit begin, okay? And there it is, okay? So you go ahead and answer these questions. You can have your browser open. You can, again, I can open this up right here. I'm going to go back into my class, okay? And so now I've got it open 2 times, okay? So we're going to go right over here to your class. There it is. So now I can do the quiz right here, and I can go over here and look at this, okay? All right, and that's the way that works. Just wanted to give you that heads up that you can do that. All right, boom, okay? So when you're done with the quiz, okay, you can just get out. I'm not gonna go into this. You save your answers along the way as you're answering it, okay? Save answer, save answer, save answer, okay? And then when you're all happy, you hit submit, okay? I should have told you about that last time, but I'm telling you about it now. Okay, so I am gonna get out of here. Leave page, okay? And now I'm just going to go back over here. Okay, so all right. So that's part 1. Remember, you bounce back and forth. So now we're going to go into the discussion, all right? And now I am really, really, really well versed in this. So I'm going to look at the second discussion. It talks about everything. And now I'm going to talk about the sequence of events that goes on to psychological stress. I perceive the stress, okay? So I see it with my 2 eyes, okay? Somebody's kind of coming at me and it's life-threatening. Boom. My brain processes that, okay? Hippocampus, okay? Amygdala, prefrontal cortex, they make a decision. The amygdala says, fire away, and it goes down to the hypothalamus. Hypothalamus interacts with my pituitary to release ACTH. ACTH goes through my blood, hits my adrenal, and releases cortisol. Now, the other thing that happens is the amygdala, through the hypothalamus, activates my adrenaline system, okay? And that's super fast acting. Boom, 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 okay? And I'm going to go over that next lecture. I'll give you an image to look at. And the 2 work together, okay? So the adrenaline is the gas pedal, and the cortisol is giving you gas, giving you fuel to do what you need to do, all right? All right, so discuss the events, how then cortisol, we've heard about what it does, okay, in terms of putting the post-it notes on your DNA, okay, the epigenetic tags, and then how that puts you kind of at risk for the disease. Alrighty? Okay, and then just, free associate down here, and that's what that's all about. Okay, all right, guys. And what I will do is I'm going to give you a couple of figures as well that will explain these kind of brain actions about the 2 limbs to the stress response. One being the fight or flight response, which is the adrenaline rush, and how the accompanying cortisol experience is also participates in all this. Okay? All right. That is it for today's lecture. Don't freak out. I know you're not biologists, but you're going to really, really learn a lot from this information and be able to apply it to your everyday lives. Okay. So with that, we're done.